And as a pharmaceutical physician with a translational medicine background, I know firsthand how critical and challenging it is to navigate the path from the preclinical research towards the clinic. And this session is all about those pivotal decisions and strategies that determine whether a program reaches patients or stalls at the bench, or as Kinga and some others called it, the valley of the death, or I like to call it lost in translation. And before we dive into the conversation, I'd like to ask each of the panel members to introduce your background and your role, and what are you bringing to the panel today?
So my name is Marin Mladinic. I'm hired by PPD. By background, I'm a board-certified toxicologist. I have almost 18 years of tox experience encompassing academia, pharma industry, and now CRO side. So I basically do everything from lead candidate selection to first in human and later stages, so up to NDA, BLA. So that's my experience.
So my name's Chris Floyd. So I'm also a pharmaceutical physician as well as Judit. My background is I spent around 15 years as a clinical academic working in clinical pharmacology, general medicine and drug development more broadly. I then recently spent 4 years at GSK within the early space, taking a number of molecules into first time in human. At the moment, I'm an executive medical director at tranScrip, which is a pharmaceutical consultancy. And there I advise early phase clients on essentially how to get into the clinic. So either as a CMO role or more as a physician to lead their project, depending on the size of the organisation.
Yeah, so hello again. My name is David Cook. I'm the CEO of Quodria. Prior to that, I worked for AstraZeneca for a thick end of 20 years in a variety of roles. I actually helped set up AstraZeneca's Systems Biology Group in 2000. I was saying earlier that was way too early. That was a complete failure. But anyway, different story. I worked in both the efficacy and safety of medicines side of things, and was also heavily involved in a big review of AstraZeneca's portfolio, which became the 5Rs paper, which got published about 10 years ago in Nature, which I was the first author. Since leaving AZ, I was CSO at Blueberry Therapeutics, and now I'm the CEO of Quodria.
Hi, my name is Joe Stanta. My background, I've been for the last nearly 15 years in leadership roles in CRO and the pharmaceutical area, mainly in the region of bioanalysis, biomarker and CDX development, and also very much on the interface of translating preclinical to early clinical and accelerated clinical development. Yeah, great to be part of this panel.
Hi everyone, my name is Debra, one of the co-founders of DotPlot along with Shefali. I have a background in product design and innovation design engineering. Did some work with the German Aerospace Center developing interactions for people with upper limb prosthetics. And now, yeah, working on DotPlot, looking to bring breast health monitoring to the home space.
Thank you very much. So to kick things off, let's ground ourselves in the earlier stages of drug discovery. What are the most critical decisions you need to make in the earlier stages of drug discovery that can either de-risk or derail clinic readiness?
Can I take that one?
Yes, please.
So I would highly urge all of the companies to think about the species selection. So basically, the animal model you're going to be used to get your product in. So analyze and do it very early. So if you use the wrong model, you're actually throwing money and animals into the wind. So very early on, you should pick the right species. Of course, different modalities have different rationales how to pick them. But for small molecules, start with metabolite identification. For large molecules, it would be more with pharmacological relevance. So that's a key thing you should have to be considering once you have your candidate. Where do we go next and into which species? Are we able to do it in these animals? If not, you are required to show it to regulatory bodies. You cannot use any species, so you might go down the route for in silico, in vitro, something else.
And Chris, I know what are you going to say, because we are coming from a very similar space. So pretend, yeah, let's just...
I'm going to surprise you then.
Okay, say something else then.
So I would say, I completely agree with your point on tox species, but I think all of the decisions you make early on can have a massive impact down the line. And probably the most important thing in my mind is defining your target product profile or your TMP. You know, if you decide that you want to do a drug for liver disease, you need to define what level of hepatotoxicity you are acceptable at the start. Because if you don't know what route you're going to take, you can suddenly end up with a load of candidates down the line which are not suitable for the disease you're after. And to give an example of how these kind of issues can cascade, if I think about CMC, and Dave, you gave a great example, if you don't make enough CMC for your preclinical toxicology, you can't go high enough. If you can't go high enough in your preclinical tox, you can't go high enough in your phase 1. You take a too low dose into your phase 2, and suddenly the best molecule in the world is failing in phase 2, all because of a decision to make 800 grams, not 1,000 grams, taken 4 years ago. There's that cascade that I think is really challenging.
Yeah, I would agree with you. I think the TPP is the single most important thing you start with. Understand what is it you're going to make, who's it for, why should they take it, how are you going to know it works? And then everything else is about working back from there through the different processes to understand all of those implications, as you say, and get back to the lab. I mean, certainly in the 5 Rs, we very much focused on then abstracting key information, writing it down. I know that sounds stupid, but understanding your assumptions with respect to what's an assumption, what's a fact, what's real, where are the questions. And I would say the general mode of action we always take is don't be scared to break your project. Ask the difficult question. You should be a bit nervous about it. You know, what's the answer going to be? Because if you fail to break it, it will move on to the next and be stronger for it.
Yeah, I think really good point. Maybe what I can add to this as well is that, you know, especially with biomarkers, focus on showing an early readout of efficacy as well. If you implement that very early on, it gives you a chance, while you might not need the data straight away, but it gives you an early chance to really start to validate that biomarker, validate in terms of not as an analytical way, but in terms of an actual usability in the disease that you're after. And that can really, and there's so many examples of that in the last 2 years, where you know, biotech companies have been bought out for like a fantastic amount of money, great purchases, because in healthy volunteers they could already show efficacy. Even before moving into phase 2, they could show the drug absolutely works and it hits that biomarker very nicely. And that's something that, again, can be sort of, you know, preloaded with regards to having the assay ready when you need it, but also start early with the development. Make sure your reagents are there, your platforms are there, and you get time to test and try and maybe also reject some of them if you don't need them, but have a think of that very early on. That's where, at the moment, especially in biotech space, where you will run out of money to run a phase 3, most likely, so you need a partner, you need someone. This is where you can show early on the value of the molecule that you have and the value. And this can be the difference between a factor of 10 in the company evaluation if you can show that very convincing data that really, you know, shows the proof very early on.
I think for us in medtech, it's really important to understand the problem that you're solving early on. Understand the problem really well. So with us, we knew that women were having a hard time understanding or differentiating what they were feeling within their breast tissue when they were doing self-checks. So what sort of technology can actually help to address that problem